Hello and welcome to this how-to video relating to the omics skin inside the SIMCA software. In this how-to video we are zooming in on the S-plots group. In general terms you can say that S-plots provide visualizations of OPLS and OPLS-DA models focusing on the loading of the predictive component, and the S-plots are there to simplify interpretation. In more detail, you can say that there are three types of plots under the S-plots group. It is the S-plot, it is the S-line plot, and it is the so-called Zeus plot. And the objective of this how-to video is to shed some more light on these three plots. In this series of how-to videos, there are two predecessing recordings. The first recording dealt with how to import a dataset, an omics dataset, into the SIMCA software and set up an omics project. And the example that we chose to use is the so-called Poplar example, which is part of the compilation of tutorial exercises that we use in our trainings. The second how-to video dealt with how to use the analysis wizard in this omics skin to calculate two models for the two-group situation. It was a principal component analysis model to get an overview of all the samples, followed up with an OPLS discriminant analysis model to separate two groups of samples, the wild type and the upregulated class. Now, in this video, we are extending the scope of this dataset a little bit. In addition to the two classes that we have used in the two earlier how-to videos, the wild type and the upregulated class, we are now also kicking alive the third group, the L5 group, which represents down-regulated samples. So in this how-to video, we have the wild type, we have the down-regulated samples from the gene in question, and we have the up-regulated samples from the gene in question. Another extension in relation to the two previous how-to videos is that in addition to the first OPLS discriminant analysis model, where we compare the wild type with the 2B, the upregulated group, we have used the analysis wizard inside the omics skin to calculate a corresponding model where we compare the wild type with the L5 or down-regulated group. So when we are illustrating these S-plots, we will use both these two OPLS discriminant models that have one predictive and one orthogonal component each. But before we go into the S-plots, let's start with a little recap of what we did in the preceding recording. In the preceding recording, we were working with the model comparing the wild type and the upregulated group, the 2B group. And we were looking at the scores, and we were seeing that this OPLS discriminant analysis model was able to locate the wild type samples in the left-hand part of the score plot and the upregulated samples in the right-hand part of this score plot. And for this model, when clicking on the loading button, the S-plot is open up. And this brings us over to a little closer scrutiny of the plots being part of the S-plots group. So the S-plot, that is a plot that is used for one OPLS model, and it is there to visualize the covariance and the correlation structure between the X variables and the predictive score. And this is a scatter plot of the P1, the loading of the predictive component, together with the correlation scaled version of itself. And this plot often takes the shape of the letter S, and that means that variables that are far outside on the wings of the S in the top right-hand part and the lower left-hand part are the best discriminators between the two classes under scrutiny. So this means that phosphoric acid and inositol and sucrose and so on are the best discriminators in the current example where we're comparing the wild type versus the upregulated 2B class. The S-line plot is very similar to the S-plot. It is an analogous line plot to this scatter plot, and it is especially suited for spectroscopy data. It is perhaps not optimal for the current situation where we have GC, MS metabolomics data. It will also visualize the covariance and the correlation structure between the X variables and the predictive score, but it is a line plot. It is again the loading of the predictor component that is plotted, but now we're using coloring according to the correlation scaled version of the predictive loading, and we're using the absolute value of the correlation scale loading. So wherever you have high peaks with an orange to intense red color, there you combine a high correlation with a high covariance. As I said, the S-line plot is most suited for spectroscopy data, so we will not look into it more in this particular video where we are dealing with GCMS data. The third member in the S-plot group is the SUS plot. SUS is an acronym for shared and unique structures, and it is a scatter plot of two loading vectors from two OPLS models. So in contrast to the S-plot and the S-line plot, in order to create a SUS plot, you will need at least two OPLS models or OPLS-DA models. And in order to illustrate the SUS plot, we have, for this dataset, created a second model where we are also involving the third group, the down-regulated L5 samples. So in addition to the first model, which was the OPLS discriminant analysis model comparing the wild type with the upregulated 2B group, we have a corresponding OPLS discriminant analysis model comparing the wild type with the down-regulated L5 group. That means that for this second OPLS discriminant analysis model, we can also create a score plot, inferring that we have the wild type in the left-hand part of the score plot, and in this case the down-regulated class on the right-hand side. We can also create this S-plot for this second model, and it also has this kind of S shape. And now in order to pave the way for the description or explanation of the SUS plot, we will have both S-plots of both models open and tile them side by side. So now here on the left hand side we have the SUS plot for the model comparing the wild type with the 2B, the upregulated group. And on the right hand side we have the S-plot when we compare the wild type with the downregulated L5 group. Now in order to create the SUS plot, we will take the correlation scale loading for both models, you can say the two y-axis in the two S-plots, and plot them together in a scatter plot, and that is when we are creating the so-called SUS plot. And in the SUS plot we will position the model comparing the wild type with the upregulated class on the x-axis, and we will place the model comparing the wild type with the downregulated class on the y-axis. And voila, here we have the SUS plot. And after tiling the two S-plots, we have them on the left-hand side on the screen. Here in the top part we have the S-plot for the first model comparing the wild type with the upregulated group, and in the bottom part we have the corresponding S-plot from the other comparison where we compare the wild type and with the downregulated group. And on the right hand side of the screen we see now the SUS plot. As I said, the SUS plot is based on plotting the correlation scale, the predictive loading for one model, model 2, against the correlation scale predictive loading for another model, model 4 in this case. So in the SUS plot, all the variables that are found on the diagonal running from the lower left-hand quadrant through the origin of the loading plot all the way to the top right-hand quadrant. Those variables are part of the so-called shared structure. It means these variables are either increasing numerically or decreasing numerically or are remaining unchanged numerically when you're comparing the wild type with the two alternative groups, the upregulated group and the downregulated group. So anything on this diagonal, that is part of the shared structure. Then, when looking at the unique structure, we would look at variable points that are located on the axis in the SUS plot, far away from the origin. So for instance, if we take the variable inositol, we can see that inositol, that is something that has a strong contribution in the OPLS discriminant analysis model, comparing the wild type with the upregulated class. So inositol is something that increases numerically when going from the wild type to the 2B samples, whereas when going from the wild type to the downregulated samples, it changes not at all, or at least not very much. So this is a unique structure. Inositol is part of the unique structure. It changes in one model but not in the other. Similarly and analogously, glucaric acid is another marker being part of the unique structure. Glucaric acid changes a lot numerically when going from the wild type to the downregulated samples, whereas it doesn't change numerically to any appreciable extent when comparing the wild type samples with the upregulated group. So in this way, by using the SUS plot, you can identify shared structures that are the same between two or more two-class models and things that are unique for a very specific two-class comparison. And of course, there are extensions to this two-dimensional SUS plot. If you have three classes you are comparing with a wild type or a reference type, or so you can use a three-dimensional SUS plot. And if you have four treatment classes or four alternative classes that you're comparing with a reference class or a wild type class, you could use a three-dimensional scatter plot and then use coloring in the fourth dimension, so to say, to represent that part. And conceivably, you could also with SIMCA illustrate a five-class comparison where the five classes are compared with the same reference class. If you have a three-dimensional SUS plot, you use colors to represent the fourth dimension or the fourth scale, and you use sizing of the plot markers to represent the fifth dimension or the fifth aspect. So, summing up, the objective of this how-to video was to illustrate and exemplify and explain the three plots being part of the S-plots group: the S-plot, the S-line plot, and the SUS plot. In a follow-up how-to video, we will look into some of the tools and functions that are available under the validation group being part of the home tab of the omics skin inside the SIMCA software. But that's another story. Thanks for watching this how-to video. I hope you find it useful.